Okay, this is Chapter 10, Part 2, and in Part 2 we're mostly talking about translation, the actual making of a protein. If you remember at the end of Part 1 video, we had our messenger RNA. It was made in the nucleus, and then it gets sent out to the cytoplasm. And out in the cytoplasm, it goes to a ribosome, and there you're going to have your transfer RNAs that will attach to an amino acid. So you have, here's your amino acid attachment site, and here's the anticodon. That's the triplet that's going to match the codons and bring the correct amino acid to make the correct sequence. The transfer RNA is all just folded up, and you can see the hydrogen bonds as it just folds over, but the anticodon site and the amino acid attachment site are the two key parts here. So each transfer RNA molecule is folded up, and what we're really concerned about, and this is just a model of it here instead of showing all the folds, we're really concerned about the amino acid attachment and that anticodon site. All right, so your ribosomes are actually made mostly of well, there's two subunits. It's mostly a protein, and there's also something called ribosomal RNA in there. And this is where the proteins, the messenger RNA is going to go, and the transfer RNA is going to bring the amino acids, and we're actually going to start building our polypeptide chain. So here in the diagram, you can see here's your messenger RNA with the codons. Your transfer RNAs, there's two of them here, and they'll be linking their amino acid to form this polypeptide chain, and that will grow out of the ribosome. You'll get a long chain, and that will fall off and fold up and become a protein. All right, so here's your, again, another model of this. This is where you're going to have your messenger RNA binding site, all right, and these are your two subunits. This is where the transfer RNA, obviously this is a model, right? This isn't actually what it looks like. Here's your two transfer RNA binding sites. Those transfer RNAs are going to bring their correct amino acid, and they will be linked together to form a polypeptide chain. So before that starts, we're going to have an initiation codon at the start of your messenger RNA, and that is always AUG. That's your start codon, and then you have an ending codon, which is, there's three of them. I think it's UAA, UAG, and UGA, I think. And that will terminate the polypeptide there. Alright, so your start codon, here it is. It codes for methionine, which means all proteins start with that because there's only one start codon, AUG. You might as well remember it because it's so common. There's the anticodon that corresponds to it, UAC. It will bring this amino acid, methionine, every single time. In the ribosome, that's going to go to the first site, and it's bringing its amino acid. And as elongation occurs, it's going to move to the next site, and the next one will move in here. Elongation just adds your amino acids to the polypeptide chain until you hit the stop codon, right? So we're just going to keep going one at a time. We'll hit a stop codon, and then it'll fall off, and you'll have your polypeptide chain. All right, so here's a nice diagram of that. Here again, you have your codons. This is the ribosome. You have the next amino acid coming in here. It's going to code here. These are going to link together. It's going to move from one site to the next site. When it's there, then the next one can scoot in behind it, and that's how you link and link and link and link and so on and so forth. Codon recognition. Here we go with the animation. All right, the messenger RNA moves one codon at a time, and the transfer RNA with the complementary anticodon pairs with each codon, adding its amino acid to the polypeptide chain. That was in the diagram you just saw in the previous slide. All right, and then elongation continues until you reach the stop codon. So here's a brief review of it. The flow of genetic material, this is sometimes known as the central dogma. It's obviously not dogmatic, but the general flow of genetic information goes from DNA to RNA to protein. Now, of course, we have found retroviruses that actually go from RNA to DNA, but science is not dogmatic, so we don't throw out the whole idea. We just modify our understanding. All right, here's your summary of transcription and translation. So here we are in the nucleus where transcription occurs. Then we move to the cytoplasm, where we're going to have translation. It's just the messenger RNA that gets sent out there, not the actual DNA. You have your transfer RNA bringing the correct amino acids. In the ribosomes, they get linked together. That's called elongation. When it finally hits a stop codon, that's termination, and the polypeptide chain, we've just created the primary structure. It's going to fall off and fold up. Now, mutations are changes in the DNA. So mutations, when we're talking about mitosis and things like that, can cause cancer. Mutations in sperm and eggs, when we're talking about meiosis, can cause genetic mutations in the child. So both of those are mutations in DNA. And it can be caused by DNA replication errors. And mutagens, something that's mutagenic, means that it causes mutations. So a lot of mutagens, you can guess, are also carcinogenic. So in this case, we have a normal hemoglobin DNA, which would code for glutamine here. And in the sickle cell hemoglobin, it codes for valine instead. So it's a point mutation, it's a single mutation, but it changes hemoglobin enough to cause some serious problems. So you can have substitutions where you just change one base pair for another. You can have insertions or deletions, and those are really bad because you will have a shift from that point on of all the base pairs after that. The triplets will be messed up. So a simple base substitution is one thing, but if you have something missing, then you'll see down here at the bottom, you see all the amino acids get shifted and they all become changed instead of just one change. That is far more severe. Okay, viral DNA can actually become part of the host chromosome. It's kind of interesting, but we are all walking around with viral DNA in us right now as we sit here. Viruses are just genes packed in protein. That's why they were so good for the Hershey-Chase experiment. But they can just stay as the DNA in us or in bacteria, both. So when a phage DNA, and remember phage is just a virus that's going to infect a bacteria, enters something called the lytic cycle inside the bacteria, it's replicated, transcribed, and translated. Okay, then you have new viral DNA and protein molecules. They assemble into new phages, which burst out of the host cell. That was the classic phage life cycle that you saw in video 1. There's another cycle called the lysogenic cycle. And in this cycle, the phage DNA inserts into the host chromosome and is passed on like the rest of the DNA. They actually inherit the viral DNA from the bacteria, from like the original mother bacteria, say. And that DNA just stays there until some trigger occurs, and it can actually go from the lysogenic to the lytic. So here you go. It can do both of these. It's kind of an interesting thing. You have the original infection here, right? So then here's the classic one I showed you, where it just the DNA gets injected, the bacteria starts making viruses, the viruses explode and go infect other bacteria, all right? Here is the lysogenic one. This one's interesting because it can just take that DNA and literally incorporate it into the DNA of the infected bacteria. And as that cell replicates, that DNA just stays in there generation after generation until some trigger where it can then go into the lytic phase. So very interesting. Two ways, two reproductive life cycles. Both of them we consider reproductive in that DNA is being reproduced. All right, so many viruses cause disease in animals when they invade an animal and a plant. Plants can get viruses as well. The flu is a very classic virus. It's actually a pretty nasty virus. We're kind of used to it, but it's pretty nasty. And the flu can have RNA rather than DNA. Right? Some animal viruses, and this is kind of tricky, steal a bit of the host cell membrane as a protective envelope. And it's kind of tricky because the host is then less likely to recognize this as an invader. And so it can remain in the body and not necessarily trigger the immune system. We have taken the DNA, the viral DNA, replicated it, and stuck it in something that our body doesn't recognize as an invader. Okay. Here we go with a plant virus. That's serious agricultural pest. There are numerous plant viruses that can cause problems, and they can get transferred. They can get transferred just from cutting shears and other things like that, wounds in the plant from the outer layer. So if you're pruning one to the next, you can actually be carrying plant viruses with you. And they invade just like they would in an animal. It's the same exact process. Okay, we have some emerging viruses that threaten human health, and what we mean by emerging is that we have recently discovered them, or they have recently come into humans, some from birds, and some from where we don't know where from. A fairly new one is Ebola, which is this one here, and that's pretty nasty, first found in Africa. And then this lovely one over here, which you think is kind of pretty until you know what it is. It's called a coronavirus because it's supposed to have this sort of halo effect, like the corona of the sun. This is actually the SARS virus, which is pretty nasty. Probably started in birds in Asia, but again, not completely sure where that one came from. All right, then the AIDS virus. Again, considered an emerging virus. It's been around your whole lifetime, but it's new to us in historic terms. And it's something called a retrovirus, which is very interesting. What it has is, instead of DNA, it has RNA. And it has something called reverse transcriptase. So it's going to, instead of making RNA from DNA, it's going to make DNA from RNA. It's going to reverse transcription. So we call it reverse transcriptase. All right, so how does that work? Inside the cell, the HIV uses its RNA template for making DNA. So we're going to go backwards. Again, reverse transcriptase is just backwards. We're going to take the viral RNA, we're going to take reverse transcriptase, and we're going to make DNA from it, all right? And that'll go to the cytoplasm, and you get the provirus, the DNA, and then that can make RNA, and then that gets packaged up and sent out of the host cell. All right, so bacteria can transfer their DNA in 3 separate ways. One way is called transformation. And in transformation, there is just a fragment of DNA in the environment from another cell. And there was actually an experiment where there were non-pathogenic bacteria, and they were put in a dish with just fragments of DNA from pathogenic bacteria. And sure enough, the DNA can enter the cell, and they could become pathogenic. So transformation is just DNA coming out of the environment. Another one is transduction, and what can happen with that is the phage, again a phage virus, that may have picked up a few fragments of DNA from another bacterial cell. This would be accidentally. It's supposed to have phage DNA, but life isn't perfect, and sometimes it'll pick up fragments of the previously infected host and then inject that into the newly infected host. Another kind is conjugation, and this is the sex between the two kinds of cells. So in this one, you're going to have a fertility factor on this one, and the sex pili here can actually attach to the female recipient, and then this DNA is copied. It doesn't donate its DNA, it actually copies it and gets a copy of that sex or fertility factor. So it doesn't matter how the DNA gets into the bacterial cell. Once it's there, it's going to be integrated into the chromosome. And the way that happens is a process called crossing over. You're going to get 2 points here, right here, yep, and right there. And this DNA is going to be removed, and the new blue DNA is going to be inserted. And this DNA just gets degraded. Whatever's left, these tails here, whatever's left gets degraded by the cell. And now you have recombinant DNA. And what that means is just recombined. It's a fancy term for recombining your DNA. But it means that this cell might have some new properties that the old cell did not have. Bacteria plasmids can serve as carriers for gene transfer, and a plasmid is just a small circular piece of DNA that is separate from the bacterial chromosome. So they'll have that regular circle. It'll also have another small circle called a plasmid, and these can transfer fairly easily to cells. So here we have ways that plasmids can work. Here you have the way we were talking about before, this integrated DNA. And here it's getting copied, and the copied piece getting into the recipient cell, and now the crossing over and the recombination. With a plasmid, what you have is the plasmid will copy itself and send in a whole new plasmid over here. And it's kind of a neat, easy way of doing it, where you don't need to have the degrading of the pieces you don't use and that kind of thing. You get the whole plasmid to come over here, and it's a circle. And now, if this is an F factor plasmid, which F factor just means fertility factor, then this cell would now become male. There are other plasmids. They're not all F factor plasmids. They can carry other genes. But in the example given here, it's an F factor. So this cell will have sex pili and other things that help it reproduce.